Welcome to this lecture on the scope of animal biotechnology. Today, we are going to explore how we can use living cells as programmable machines to solve some of the world's biggest problems. Whether you are completely new to this topic or looking to deepen your understanding, we will build everything from the ground up. By the end of this session, you will not only understand the biological concepts, but also the mathematics behind scaling up these incredible microscopic factories. Let us dive right in. To begin, I want you to imagine that you have a tiny, microscopic factory. This factory can build exactly what you need, whether it is a life-saving medicine or a nutrient-rich food. For thousands of years, humans have unknowingly used these biological factories, like using yeast to bake bread. But today, we do not just rely on chance. We actively program these living factories. This is the very heart of biotechnology. By understanding how to read and write the genetic code of life, we can instruct animals and their cells to manufacture things that were previously impossible to create. Now that we have an intuitive feel, let us look at the formal definition. Biotechnology is the application of scientific and engineering principles to the processing of materials by biological agents. In animal biotechnology, this means using animals, their cells, or their genetic components to create beneficial products. Every word here matters. Scientific principles refers to our deep understanding of genetics and molecular biology. Engineering refers to the precise tools we use to physically alter the DNA. Ultimately, we're bridging the gap between raw biological potential and practical real-world applications, like introducing disease resistance or producing therapeutic proteins. Building on that definition, let us explore why this field is so incredibly important. The scope of animal biotechnology touches almost every aspect of modern human life. In medicine, it allows us to produce life-saving drugs, such as recombinant human insulin, directly within the milk of transgenic animals. Without this, we would still rely on extracting insulin from slaughtered pigs, which was highly inefficient and caused severe allergic reactions. In agriculture, it helps us breed animals that grow faster and resist devastating diseases. By expanding what biological systems can do, we are moving toward a future with cheaper medicine and abundant food. To visualize what we just discussed, let us look at this diagram illustrating the three main pillars of animal biotechnology. In the center, we have animal biotechnology, branching out. On the left, the blue section represents medicine, showing a bioreactor and a vial of recombinant proteins. On the right, the purple section represents research, featuring a DNA helix and a laboratory mouse used as a knockout model for disease study. Finally, at the bottom, the green section represents agriculture, showing a transgenic cow. This visual perfectly summarizes how manipulating genetic codes translates into powerful industrial, medical, and agricultural tools. Having seen the broad applications, we must now ask, how do we actually manufacture these products on a global scale? To truly understand the industrial scope of biotechnology, we must understand how we scale up production from a single cell to a massive factory. When we engineer animal cells to produce a medicine, a few cells are not enough. We need billions of them. We grow these engineered cells in large, carefully controlled tanks called bioreactors. Inside these tanks, the cells multiply exponentially over time. By using mathematical models to track this growth, scientists can predict exactly when to harvest the cells. This brings us to the mathematical model that tracks this exponential multiplication. The formula is n of t equals n0 times e raised to the power of r times t. Let us break down every symbol. N of t is the total number of cells at any given time t, measured in hours. N0 is the initial number of cells we start with in our bioreactor. The constant e is the base of the natural logarithm, about 2.718, which naturally describes continuous biological growth. The variable r is the specific growth rate, telling us how fast cells divide per hour. If the growth rate r increases, the total cell count explodes rapidly, speeding up production. Now that we have our formula, let us see this with a concrete example. A company starts with an initial population of 1,000 cells. The specific growth rate is 0.05 per hour. We want to find the total cells after 48 hours. First, we identify our knowns and plug them into our exponential growth equation. We replace n0 with 1,000, r with 0.05, and t with 48. Multiplying the growth rate by the time gives an exponent of 2.4. Evaluating e to the power of 2.4 gives approximately 11.023. Multiplying this by our initial 1,000 cells gives a final answer of approximately 11,023 cells. Physically, this means our microscopic factory has grown 11 times larger in just 2 days. So we have successfully grown our cells and proven that exponential growth works. This brings us to an important question. How much actual medicine do these billions of cells produce? It is not enough to just have the cells. We need to quantify the therapeutic protein they manufacture. This calculation is fundamental in the scope of industrial biotechnology to ensure a manufacturing process is economically viable. To do this, we use a simple linear production model. For this specific production phase, we assume the cell count remains stable, allowing us to calculate the total mass of the protein produced over a set period. Let us look at the mathematical form of this concept. The formula is m total equals n cells times p rate times t. Here, m total is the total mass of the protein produced, measured in grams. N cells represents the total number of cells currently active in the bioreactor. P rate is the production rate per individual cell, usually a very tiny number measured in grams per hour. Finally, T is the total time allowed for production, in hours. Conceptually, if you increase the number of cells, or the time allowed, your total yield increases linearly. This simple multiplication allows biotechnologists to predict exactly how much life-saving drug they will harvest. Before we move on, let us apply this formula to a real scenario. Suppose our bioreactor contains 50,000 transgenic cells. Each cell produces 2 × 10⁻⁹ grams of protein per hour. We want the total mass produced in 24 hours. We substitute our known values directly into the production equation. First, we multiply the 50,000 cells by the microscopic production rate of a single cell. This tells us that the entire bioreactor manufactures 0.0001 grams every single hour. Next, we multiply this hourly rate by the 24 hours in a day. Our final answer is 0.0024 grams, or 2.4 milligrams. While tiny, biological drugs are incredibly potent, proving how we can optimize yields from microscopic factories. We have covered a lot of ground today, so let us reinforce the most important ideas. First, animal biotechnology is transformative. It turns basic biological processes into powerful industrial, medical, and agricultural tools by manipulating the genetic code. Second, we saw how mathematical models, like exponential cell growth and linear protein yield, allow us to scale up production from a single cell to a massive bioreactor to meet global demands. Finally, remember that biotechnology is not just about studying life, it is about actively partnering with life to solve the most pressing challenges of our modern world. You now have a solid foundation in the scope of animal biotechnology.